Hello, everyone, and welcome to our 2024 PKD Summit. My name is Michelle Lynn Goodfellow, and I'm the Communications and Engagement Manager for the PKD Foundation of Canada, which is the only national organization solely dedicated to fighting PKD through research, education, advocacy, support, and awareness. In this session, our 2024 PKD Research Grant recipient, Dr. Marie Trudel, will be presenting her team's latest preclinical research findings on a key ion channel's role in polycystic kidney disease. This presentation, designed for PKD patients, their families, and friends, will cover the basics of PKD, how kidney cysts form and grow, and the current treatments available. Dr. Trudel will explain how researchers are identifying the main factors driving PKD and how they've discovered an ion channel that can be controlled with a drug to potentially stop the disease. She'll also share exciting updates about her ongoing work on liver cysts and new ways to target polycystic liver disease, or PLD, for future treatments. We'll be leaving time for a live question and answer session after Dr. Trudel's presentation. So feel free to leave your questions either in the Q&A area or in the chat throughout the presentation, and we will try and address them at the end of the session. This session is being recorded, and it will be available to view on our YouTube channel in the coming weeks. So I'll now introduce our panelist. Dr. Marie Trudel is a full professor and director of molecular genetic and development, a professor in the Department of Medicine at Université Montréal, and an adjunct professor at McGill University. She did her PhD in biochemistry at Institut de Biologie Physico-Chimique in Paris, and her postdoctoral fellowship in DNA damage and repair at the Imperial Cancer Research Fund in London, England, and her postdoctoral fellowship at Columbia University in New York. In the last 30 years, the Trudel Laboratory has established a major research program on the study of ADPKD. The lab aims to decipher the cellular and mechanistic function of gene responsible for ADPKD. They have developed unique and highly pertinent mouse models of PKD that most closely reproduce human ADPKD, with not only the renal disorder, but also several types of the typical extrarenal phenotypes, including liver cysts, cardiovascular anomalies, and cerebral aneurysms. Studies of these mouse models have provided clues on key signaling pathways and molecular effectors using genetic, cell, biological, biochemical, and multi-osmic approaches. Her lab pioneer work on the regulation of PKD1 has also determined the roles of different transcription factors and epigenetic mechanisms. Studies from her team have advanced our understanding of the molecular pathogenesis of ADPKD and identified potential therapeutic targets for PKD treatment toward clinical investigation. Dr. Trudel has received numerous funds from the CIRH on a continuous basis from 1989, from the NIH, DoD, and the PKD Foundation of Canada, the PKD Foundation in the US. Dr. Trudel participates actively in the PKD Foundation USA Scientific Advisory Board, in community services, educational and awareness programs, and she has also organized several conferences, been involved on various boards, been a member or a chair jury of committees, and served on grant and scholarship committees. So we welcome you, Dr. Trudel, and we are looking very forward to hearing what you're going to share with us today.
Thank you very much. Very good afternoon, everyone. I thank you very much for the invitation to present our work today. Our study spans from the very basic or fundamental mechanism of ADPKD, so hardcore scientist, but I also lend a hand and go towards more translational understanding to develop therapies. And today I'd like to speak to you principally about a discovery we made that could have promises for renal cysts. At least for the moment, that's what we know. I'll try to be clear, and if there's question, please excuse me if I use inadvertently a scientific term. I will try to catch myself up if I do that. But I know that some of the PKD patients are extremely knowledgeable, which is really something that I must say as a research scientist is really pleasing to, and it's an honor for us to have such encouraging patient to work for. So I know some of you must know part of this, but I will go through it again. My lab is interested in autosomal dominant polycystic kidney disease. It's probably the most prevalent genetic disease from a monogenic point of view. It affects 12.5 million people in the world and 60,000 people in Canada. Because it's an autosomal dominant form, if one of your parents have this disease, you're liable to obtain this disease at 50%, the probabilities of ADPKD. And it has stayed in the population at such a high rate that 1 person in about 500, there's discussion about the literature if it's 1 in 500, 1 in 600, or many discussion, but it's a very high rate. And it is irrespective of sex, racial, or ethnic origin. And so it stayed very prevalent in the population, especially because parents usually discover that they have the disease usually once they have had their children. So the kidney usually is big as a cyst, as a fist. And ADPKD kidneys enlarge as big as a football ball by having so many cysts that covers them. This growth affects the function and causes a lot of discomfort. The kidney can fill up a lot of the body cavity, and it's crunching all other organs, and this compression can cause additional damage. So what do we know about the kidney? The kidney leads often at about 50% of patients in midlife will require either dialysis or transplantation. Associated with the symptoms of ADPKD are often hypertension. I just talked about the enlarged kidneys, kidney stones, renal infection, blood in the urine, and there's also quite a few extrarenal manifestations. Perhaps the most prevalent after the kidney is the liver. And the more we're studying the liver, the more we see that it's something we had not focused as much and maybe had not noticed the presence of cysts in some patients with ADPKD. There's also other extrarenal manifestations that are pretty bad, and such as cerebral aneurysm, cardiac valve anomalies, but there are much more and a lower proportion in patients with the ADPKD. In this slide, I always talk about the cost of ADPKD. It's enormous to treat people with ADPKD, so we have everything to gain to find a cure or to find something that delays this progression. And this is for the politician amongst you. It's important to invest in ADPKD. So, what is cysts? As you can see in this, maybe I could point, cysts at a very early age show this. These are kidneys, by the way, and that we can see in function of age. And you can see that cysts start at very early age and start to be filled up with fluid, and it grows with time due to hyperfiltration or trying to compensate. And the cyst grows so much in size and in numbers that they compress the other good tissue and destroy the kidney. So two functions are readily apparent that even in the early stage of studies of ADPKD is that the kidneys undergo a lot of cell division or proliferation or enlargement, and they have accumulation of fluid and secretion of fluid. So we know these are the two principal characteristics. So what do we know also about the genetics of ADPKD? Amongst the disease, I hinted to this, PKD, there's a gene called PKD1, which is the most prevalently mutated in ADPKD. 85% of the cases are approximately or have mutation in PKD1. Actually, in contrast to other genetic disease, almost each family pedigree has their own mutation. So there's something unique about it, and that makes mutation throughout this gene. At present, we have at least characterized among the researchers more than 2,000 different mutations. In our study, it encodes a protein, a very large protein that maybe you can see behind me, called polycystin, which we've done studies, but I won't speak too much about it today. But polycystin is a very large protein of unknown function. And so our study mainly focuses on PKD1 because it is the most prevalent, and it is the one that caused the most detrimental effect in the kidney. But it's also a very big challenge because it is very difficult to study such a big protein. There's also a second gene that has been identified, and others that develops a little bit later than those affected by PKD1. The trend is that. So our belief is that knowledge of ADPKD from studies on mouse models will allow us to understand the biologic processes that control cysts in ADPKD. And this could lead us to develop a rationale for more efficient therapies. So this has been the motto in my lab, understanding who we should target, how we should target, when we should target. And part of this is what I'm going to try to talk to you about today. So for those of you that may not know, a kidney has this bean shape, or the bean has a kidney shape, and is full of a long tube that we call nephron in the scientific or biomedical language. And this is the unit device for filtering. In our kidneys, we have about 1 million of these nephrons. And actually, there are in excess, because we just need 10 to 20% of our nephron to be functional, to have a kidney that suffices to our well-being. These nephron or tubes, they filter at least 200 liters of blood per day, which is enormous. So what happens in ADPKD is this tube that you can see here schematized have cells that are mutated, for example, as shown in blue here. And these cells can undergo proliferation. These are single cells that undergo proliferation or subdivision or duplicate themselves. And they secrete fluid. And fluid will accumulate to form a cyst when it detaches from the main tube. If we look in a kidney of an ADPKD patient, people have seen that there's about 1,000 cysts, a couple 2,000 or 3,000 cysts. So this should leave us, in theory, with 999,000 nephrons that are good, because the cysts affect only one nephron. But this tells us that one cyst disturbs all the other normal nephrons. So I'm not sure you can see here at the bottom, but there was a question that was, what are the factors that are responsible for this? And could we possibly find therapies and targets for this formation of the cysts? So what are our current knowledge? Our current pharmacological intervention is tolvaptan. Tolvaptan or Gernac had many other names previously, but it's the only drug approved for ADPKD. And its function is to block the vasopressin receptor. I know it's a mouthful, but it is a receptor at the bottom here of a single cell that you can see here, which I illustrated to you previously in blue. This molecule is secreted in very big quantity in ADPKD because it senses that the urine is not concentrated enough. And it sends a signal to the cell to concentrate it. And it activates the vasopressin receptor. So this is like a lock in the key. And tolvaptan is blocking vasopressin from activating this receptor or this lock. And so the whole system that is usually triggers a chain reaction, which starts with this interaction of chain and of lock and key, is blocked by tolvaptan. What we know about the side effect of tolvaptan is that it causes a lot of urine output. And also because of the very logically a lot of urine output gives a lot of thirst, and people drink a lot. One side effect of tolvaptan is toxicity to the liver. And not everyone has toxicity to the liver, but it does affect. And the company that make tolvaptan is also very aware and take care that the liver toxicity doesn't develop in patients that receive tolvaptan. There's also drug interaction. So as well, what we know as another interesting phenomena is that there's a high proportion of non-responder. And this might be because there's a poor therapeutic window. So let me explain to you what I mean by a poor therapeutic window, although some of you might know, is that you need sufficient tolvaptan to block this phenomenon. But if you go overboard, you might have liver toxicity. So we do have a very small range in which it may vary from individual to individual and may not allow it to block this function of tolvaptan and make them non-responder. The other caveat maybe is that there's no vasopressin or vasopressin receptor in the liver. So it's not a treatment for liver cysts. All in all, there's probably room for improvement. And it's based on this that we started a certain reasoning on it. What we know, we reason that if the mechanism of tolvaptan is to slow down cysts, and it is working, perhaps we could exploit this mechanism a bit further. So I will try to, this is maybe the most difficult slide I have from the mechanistic point of view. Because I will try to explain to you how we came to this. So what we know is it possible to remove this and myself almost so that we can see this slide?
Hi, Dr. Trudel. It has to do with the settings when you shared your slides.
Okay.
So you might want, if you do want to try and fix it, you would have to stop sharing. And then when you want to go to share again, choose a different setting so that it's just,
This is
it's
maybe
just,
difficult for me technically.
okay.
So, I'll try to push on one side or the other. So as I told you, underneath here, if you remember, there's the lock and key of the vasopressin. And this gets activated normally. And when it gets activated, it stimulates another receptor called the cystic fibrosis receptor, or CFTR. And when this ion channel gets activated, it secretes an ion called a chloride ion. And this goes along with water. And it goes into the tube, which I told you before. And as it does this, there's more secretion of water, there's more secretion of chloride. And you will get a cyst that will start to form and cause enlargement. We believe from studies in vitro or in culture that this CFTR channel is extremely important. And we think that that's how the whole system is working. But we don't think that we can block totally the CFTR channel. If we block the CFTR channel, the individual will be liable to develop either a pancreatic or bronchitis problem, actually the typical cystic fibrosis problem. So changing ADPKD for cystic fibrosis is not a good exchange. But unfortunately, there's some patient that inherited of both CFTR and a PKD1 mutation, an ADPKD, and these patients have a much less severe cystic phenotype. So this supports that CFTR, this channel, which secretes the chloride channel, the chloride ion, is very important. But the cell, to bring you in the mechanism, if you secrete a lot of one ion that you need, the cell, to get an equilibrium, will want to gain more chloride to compensate within the cell. So to do that, it will, in a chain reaction, ask another ion to pick up some chloride ion to go into the cell. When doing that, they're supplying the CFTR channel, if you understand well. When they pick up chloride ion, they pick up two other ions. This one is sodium, and sodium can be eliminated. How will I move? Where will I go? To the right, to the left? Okay, if I remove the sodium, then there's another channel that will remove this sodium, but in exchange, will pick up a potassium. You can forget partially this, but it is very important. The potassium being picked up and the other potassium that's here become in excess into the cell. And because of that, we need probably another channel to get rid of them. So two things. One, the sodium potassium, this channel and this channel, the NKCC1 and the sodium potassium ATPase, you can forget their name. We cannot target them as an inhibitor or a drug because they're very important in the brain. So we cannot use those. But what is interesting is that by activating the KCNN4, we're having a vicious circle here. Each time we're activating CFTR, we're activating these two other channels, and perhaps this third one that has been overlooked, and we're triggering a vicious circle. So if you follow me up until now, and if I try to be clear, if we target potassium channel, this potassium channel called KCNN4, which is the key target that have not been studied very much, maybe there's something we could block here. So when we started these studies, we knew that KCNN4 was induced in cellular stress condition. And we thought, well, ADPKD certainly, or PKD1 mutation and PKD2 mutation induces stress in cells. So perhaps KCNN4 or this channel is important. We knew from the literature that it regulates the cell volume. And we knew as well that it has function that are, it is implicated in function which are abnormal in ADPKD, which is proliferation. I talked to you about division and duplication of cells. In the fibrosis, if you have heard, which means it's something in the tissue that is causing damage, and apoptosis. So because of that, and because of our knowledge that there was a drug against KCNN4 for another disease that was available and could be perhaps used for ADPKD, we question whether KCNN4 could be important and play a role in ADPKD. So the first thing, we then investigated whether KCNN4 could be implicated in human and mouse ADPKDs, kidneys. So for this, we measured the level of KCNN4 in human ADPKD kidneys. So as you can see here from the in the color of ADPKD, in the famous still, you can see that the 9 human kidneys that we had elevated levels of KCNN4 compared to normal kidneys. I wish to remind you here that all these 9 human ADPKD kidneys have different mutation because they all don't come from the same family. So it did say that it was kind of a general phenomenon. Then we used some of our mouse models that we have of PKD1, but different mutation, 4 different type of mutations and defect in the PKD1. And you can see that it is elevated, KCNN4, in all of our mutants. So with this in mind, we decided to establish an experimental approach that we could use. So from the human that we know different mutation, we produce or model this in mice. We model 4 different type of mutation, as you saw in the previous slide. And we decided to take the kidney of these mice and to test them in organ culture, which could be relatively rapid, and to test in the mice the response of our studies with KCNN4. And then if we did have a positive response, we can hope to go back to human. So with this in mind, our first set of studies was to look at the a mouse kidney, which is nice burgundy. And as you can see here, this is like a longitudinal look But if you cut vertically, this is what you will see. So in a normal kidney organ, you will see no cysts, and they're normal and beautiful. If you take a mutant kidney from a mouse, and you put it in culture at the same time as this mice, they do produce a lot of cysts, numerously and in size. So at this point, we knew we could use this as a tool to evaluate the response to treatment. So our first experiment, or set of experiment, was to see if KCNN4 could be stimulated in normal kidney and in a kidney that is mutated for ADPKD. So the KCNN4 stimulator at higher levels starts to get cysts in a normal kidney. It becomes much more prevalent if you increase the level of the stimulator. So even a normal kidney can develop cysts. In the mutant kidney, it will have cysts normally, but it also stimulates or exacerbates the cysts further in the number and in size. So higher the KCNN4 is, the more you have cysts. So this was quantified and analyzed thoroughly, but I skipped the analysis for the quantification. So the other question now is this KCNN4 could be safe. So as a primary analysis for the following treatment, we question, can these this is a normal kidney from a normal mice. But if you remove in the mice KCNN4, the kidney looks intact. In fact, a mice that don't have KCNN4 it's in its entire body is not distinguishable from a normal mice. It is healthy. The only difference, it has a mild alteration in the red blood cell volume, as I mentioned to you before. That's the only difference. So this tells us that a mice that has no problem or is normal, and you remove KCNN4, has no issue. So the concept is that it's only important maybe under stress under stress condition. So this observation is positive and suggests that if we inhibit or remove KCNN4, it could be well tolerated in the kidney and even in the entire body, which is important for the patient. So we then went and removed KCNN4 in a mutant mice. So here you will see a normal mice, which has no cysts, mutant kidney, a mutant normal kidney of a normal mice, and a mutant kidney from a mutant mice. And if you remove KCNN4, we have no cysts, or virtually no cysts. So the cysts are reduced at in very high proportion. So the quantification says 90%. So we then question in another set of experiment, if we look in the mutant mice, provide no drug, you see cysts. Then we tried the drug that had been used in another disease here, which are called the drug inhibitor for not to have a mouthful. And you can see that when you use the inhibitor, the number of cysts, the surface of the cysts is very much decreased, and the cysts numbers are decreased, you can see by your own eyes, and so the drug appear efficient. So then in another strategy, to be closer to human, because in a human situation, when we give the drug, the patient probably would have developed already the cyst. So using the same approach as we did for the culture, we let the kidney culture develop for 3 days and develop cysts, as you can see here. But then on day 3, we did not add the drug, and you can see that the cysts continue to progress in number and size. But when you put the inhibitor in a very short time, we were expecting the cysts to stay at the same level, but we see cysts almost disappearing, reversing, as you can see at step 5. Below is illustrated the calculation of the cysts. In black are those that didn't have an inhibitor, and so at day 3, they had cysts, and they continue to grow. At day 3, of those that were mutated and received the inhibitor, it goes down in red here. So these experiments were very encouraging, and it led us to go into the mouse model. So in the mouse model, we removed KCNN4 in early onset ADPKD mouse model. And you can see when we remove the KCNN4, the kidneys are smaller. You can see that in these mice, there's an enormous amount of cysts. And without the KCNN4, there is still cysts. But some of the kidney tissue are preserved, but it's much less prevalent than in the naturally occurring mice. So it sounds very positive, and from the body of the mice was fine. So we then did the same experiment in an adult onset, which is closer to mimicking what's happening in the human situation. So you can see that the kidneys are smaller. Without the KCNN4, there's less cysts. I will try to go slowly because it's a very crowded slide here. You can see that the light blue, the cysts in different tubules are going lower. The fibrosis is going lower. The proliferation or the cell division is decreasing. What is important in this adult onset, we were able to measure the function of the kidney, and the function of the kidney is more preserved. It is not perfect, but much better, and there's a greater improvement in the kidney function. And in these mice, we can see, we measured the survival of these mice, and they survive 40% longer. They have an extended lifespan of 40%, which, as you can imagine, would have enormous impact if this could be true in humans. So we then went to the pharmacological study. Here I'm going to show you only one of the 2 different models we use in the early onset. Giving the drug for only 10 days to these mice allowed them to preserve quite a bit of the kidney. You can see here the cysts are lower, and you can see, because I'm there, that the fibrosis, where can I put myself? Decreases to an extremely low level in these mice. So we did the same thing in the adult onset mouse model, and we had 2 different adult onset mouse models. We gave the drug for 12 weeks between 6 and 18 weeks of age. You can see that the cysts at very low dose is decreased, and at a higher dose, and we could go almost 10-fold higher this dose without being toxic, prevents almost cyst formation. And you can see with the 2 doses, the decrease in cysts and the decrease in fibrosis. Oh boy. And this is the function of the kidney, which almost comes to normal. So in summary, in human, KCNN4 is very high. In organ culture, the KCNN4, if you stimulate KCNN4, you have more cysts. If you inhibit, pharmacologically with the drug and with the genetic, we repeat a decrease in the amount of cysts, and in organ culture, we even have regression. In the mouse model, we also have increased KCNN4, and you can suppress the PKD phenotype and even extend the lifespan using the genetic of in the mouse model. So this target, KCNN4, and its drug is very promising as a therapeutic target for preclinical investigation. So in conclusion, assuming that our finding in the mice can be transposed to human, and I always be careful to go too far because a human is not a mouse. Cinecapoc, which is the inhibitor, should postpone appearance of clinical symptoms by up to 40 years, which will prevent kidney dialysis and transplantation in most patients and improve quality of life for ADPKD patients. So if this holds true, we're now going to clinical trials. Why do we have this rationale? Well, our studies I presented to you. Previous studies on this drug has been done for the toxicology, the cardiogenicity in rat, donkey, and mouse. Even they're non-toxic, non-carcinogenic. They do not affect reproduction. It has no genetic toxicity. The amount of safety of this drug is amazing. This drug has already been studied in phase 1 and partly in phase 2. So it may have a high clinical impact for patients with ADPKD. So we know this from this drug or inhibitor called cinecapoc that was used in sickle cell patients. It went into phase 3. We know it inhibits 90% of KCNN4 activity. We know it is efficient, and patients have responded positively because we know how these patients have responded. And a key issue is that patients with sickle cell have often a subclinical nephropathy. And none of these patients develop hypertension after a year and a half treatment. So for us, there's no other drug with such safety and efficacy profile in animal models and even in human with sickle cell. So our future plans are to initiate a pilot clinical trial to evaluate the dose and the pharmacokinetic. Although they have been done before, they have not been done in ADPKD. So we will test this a priori as the first thing. And we could even evaluate the compliance by looking at the red blood cell. How simple a test can one hope for? And then we hope that we can do a large random clinical trial following this. At the present time, we're trying to get the cinecapoc produced in a large batch. We have a team that we're trying to complete. We're hoping to have a non-objection letter for Halt Canada to have this approval, and the FDA in the US with the IND for the KCNN4. Maybe this is too thematic, but it's trying to going forward towards a clinical trial. And must I say, we need funds to do all of this, which we need to do. Let me say 2 words about liver cysts. I told you before at the very beginning that there's extrarenal manifestation. And one of the most prevalent is the liver cyst and biliary cyst. It has been thought for a long time that it has a similar pathogenetic mechanism. Let me call it differently. The same way it is working in the kidney that it is working in the liver. But our studies point that this might not be the case. And we are questioning, how is it working? First, there's no vasopressin, so it can't work the same way totally. And we know it's very prevalent in human. And as you can see here, this is a normal liver, and this is an ADPKD liver studded by cysts. I will not mention these other extrarenal manifestations, although my mice have them. We have two hands, and I go to the most important, which is the liver cysts. So we did develop mouse models that developed, this is a normal liver. This is ADPKD mouse that has liver cysts, as you can see, just from the exterior, it's almost translucent. So we made several mouse models. One of them I'm showing to you that develops at young age. At 1 month of age in a mouse, you can see the presence of cysts. And at 3 months of age, it's also much more severe, and it's almost looking like a lace. It's so impressive. We also made another mouse model, which I think you'll have difficulty to see, but maybe you can see here at 6 months of age, it's starting to look like the one at P30, at 1 month of age. They have clusters of cysts. And at 1 year of age, they have many, many cysts. So what we observe is that the type of mutation might influence the liver cysts and can lead to a different frequency of cysts in the liver. And we're studying this mechanism, and we're hoping to be able to identify targets and to be able to correct it by developing drugs against these targets. We have clues. It's a bit early. These studies are ongoing, and we hope to eventually find a mean to correct or stop these liver cysts. In closing, I'd like to acknowledge people that have done the work on KCNN4, Seth Alper, a collaborator that is in Boston at Harvard, Almira Kurbegovic, that has been working as a research associate for close to 20 years with me, 2 students, Guan Hanyao and William Foley, a postdoctoral fellow, Camila Parot, and students that came as summer students, most of them, helping us with these studies. I'm also extremely grateful to the PKD Foundation for their support. I'm sorry, the Canadian Institute of Health Research, the American Department of Defense, the Allen Foundation of the US, and also extremely grateful to our mouse models that are helping us translating our basic finding to the patient. And if you have any questions, I'd be happy to answer them. And I hope I didn't went over too much.
That's okay. As long as you have time, we can answer as many questions as we want. Would you like to stop sharing your presentation, and then the screen behind you will disappear?
Okay, I stopped sharing. So what do? I stop sharing.
You should see sort of at the top center.
Yeah.
There we go. Yes. Very good. All right. Number one, thank you so much.
I don't see you, but that's okay.
You don't see me? Oh.
No problem. It's a problem because I could see you up, but I cannot see you down.
Yeah.
That's okay.
Well, I understand. So we do have some questions. I will start with the ones that are in the Q&A area first, and then I'll move to some of the questions that have come in the chat section. So our first question is, when liver cysts are the result of ADPKD, would a treatment that's effective in the kidneys necessarily be effective in the liver? And I know towards the end of your presentation, you did start to talk about liver, but if you would like to sort of reiterate that for that person.
Yes. it would be lovely we find a common pathway that is found in both. We always thought that it was the same mechanism, but I think there's some particularities to the liver that are different from the human. But I must say that I have not tested even the drug that I talked to you about the kidney, the KCNN4, how it is in the liver, which it's a shame, but we didn't have time yet. And it could, because KCNN4 is independent from the tolvaptan lock and key mechanism I said before. So it could be implicated in the secretion of fluid in the liver, but I cannot address this at the present time.
Okay, thank you so much. Our next question is, in humans, there are many compensatory pathways. If you inhibit KCNN4, are there any potential compensatory pathways to watch for that might be stimulated in ADPKD kidneys?
Possibly because I know that in the mice, KCNN4 does not totally block the appearance of cysts. However, I didn't talk today because I thought I had said quite a bit. I looked at different channels in combination with the drug of KCNN4. And we do have complementary and much more, how do you say, synergistic or powerful response when you have two different drugs.
Okay. Someone's asking, does tolvaptan work better at a certain stage of GFR?
To my knowledge of the literature, it works at almost every stage, or and so I don't know. I don't know if it, and it does raise the question, would it work better in different type of mutation? I don't know either. I don't think it was never addressed in the literature.
The last question that I see in the Q&A section is actually a comment from one of the people who is watching, Louisa, who is saying, thank you so very much for your dedication and compassion for patients who are affected by both ADPKD and ADPLD. You are giving hope for our children who unfortunately inherited our disease. So some wonderful thanks that we also echo there. We do have some questions.
Thank you so much, Louisa, and everyone.
We do have some questions that people have put in the chat, so I will go
Yes.
through those now.
Yes.
Someone's asking, if you don't have cysts by the time that you are 30 years of age or older, does that mean that you don't have the gene for PKD? Or at what age can someone be sure that they don't have PKD, that they have not inherited it from their parents?
I'll try to start by the end and come back to the question. It is possible for a normal individual that don't have a PKD 1, 2 mutation or other genes that have been discovered more recently to have cysts in the kidney. Serendipity or presence of cysts in the kidney have been seen in people that don't have genetic disease. Usually, cysts appear prior or 20 years of age. And there are some definitive criteria of the two kidneys being affected, the number of cysts number of cysts that are found in the kidneys to be called an ADPKD patient. This is defined previously, but usually if you have absolutely no cysts at 30 years of age, the probability of you having ADPKD or close to nil.
Okay, great. Someone is asking, is Senecapot hard on the liver, meaning that those with PLD and PKD wouldn't be able to use it? And I know that question was asked before you started talking about research into PLD, but if maybe you want to speak to that.
What we know is that the dose that we use for Senecapot is 120 milligram per kilo per day. And we know that it has been given to, I'll say, healthy individual, put it this way, at 1,000 milligram, so more than tenfold the quantity I put, and it was still not toxic. But I'll put, we say in French a bémol, like a tone down on this, because this was done in normal individual. So we know it's not toxic in a normal individual. I'm very prudent. So I'm going to tell you, we don't know for ADPKD because I didn't went to those quantity in the mouse. If not necessary to cure, why should we go to the maximum? If curing requires less, and theoretically, we had to put much more in the mouse than we would put in human. Because in the mouse, the metabolism of Senecapot is rapidly destroyed, while in human, we would need like a 10 milligram per kilo per day. So we would be 100-fold below what has been given and that we know was not toxic. So I do not believe that it would be, but I don't have the definitive answer.
Okay, thank you very much. The last question in the chat, someone is asking, how long does it take for clinical trials? And when do you expect that this particular drug would be commercially available for treatment of people with PKD if it were found to be an effective treatment?
Well, unfortunately, the system in which we are, everything is very slow. And I do understand that we need safety guard, and that we need administrative approval. And it takes a very long time to get administrative approval, close to almost a year from Canada and the US, many months. So the steps are extremely long, and if I base myself on tolvaptan, it took many years before tolvaptan was approved to be given to patient. I don't know the exact number of years, but it certainly was above 5, I would say closer to 7 to 8 years for the entire population to get. And you need approval in each country. But the clinical trial, I would expect, I'd base myself, or we've based ourselves for going into the clinical trial on the experiment of tolvaptan. So I think we would need a 2 years follow-up, if not more, to be able to see the positive effect. So that maybe answer partially the question of the person, but before it is on the market, I'm not sure how long it would take.
I understand. And there has been another question come in, which is very related to that last one. Any estimate when human clinical trials might start? And how, if one wanted to participate in the research, how they would apply to become a participant?
Well, I don't know, but I'm saying this off the top of my head without ever consulting anybody. But we are in the process of doing it, and we are in the process of doing all the papers to get everything ready and to get the drug ready. So I certainly will try to advertise that we're doing a clinical trial. But if the PKD Foundation of Canada would like to help me in advertising that we will be doing this, then I'd be delighted. You could go to the site of PKD Foundation, but I cannot say this without, I've not even spoke to them about it. So I will wait to do the first step before I go further. But if it goes in and it's approved by Health Canada, I will try to get this advertised as much as I can within the community, perhaps via Luisa also. I'll do my best so that the word spread.
And I know that you are based in Quebec, in Canada, would other patients across Canada be eligible, or do they have to be local to you if the trial were to go ahead?
Pilot trial, I probably will do it in Montreal and in Boston, where my collaborator is. So we'll be two small centers, or two centers. I can't say over the small center because I hope you didn't record this. Two centers that will do the small pilot essay. But when it comes to the large randomized clinical trial, I will try to tap on every clinician that is interested in ADPKD in Canada to want to participate. I hope we'll have, the more we have participants, the faster we'll have the result and we'll know where to stand. And so I will rely on colleagues and clinical researcher to help us.
Thank you. Well, and without official confirmation, I can say that the PKD Foundation of Canada is really committed to our community and to spreading information that will help people affected by PKD in Canada and beyond. So we are always happy to spread information that will be helpful.
Thank you very much. Thank you very much. And I'm really hopeful it can go further.
We have someone in the Q&A has said, I am ready to help out now. So
thank you very much. And I'm sorry I went a bit overboard, and I hope I didn't use too much scientific terms.
That's okay. Are you all right for time? We did have a couple of more questions that came in from registrants ahead of time. Do you have a little bit more time to answer those?
Absolutely.
Okay, I will just bring those up. And many of them you have already answered in your presentation or in the question and answer period. But someone is asking, would your research findings affect ARPKD patients as well as ADPKD patients in the future? Is that something that is possible?
It is quite possible. It is quite possible because several of the things that we've done, I think would, but we need to do the study very carefully. I didn't go directly there because I think that usually in clinical trials, they start in adult individual to give a new drug instead of an infant. And ARPKD usually is in infancy. And we need to, our studies did use very young, growing, developing mice, baby mice, because I gave it to them when they were 5 to 10 days old. And so we followed if the drug had an impact on their weight, on the toxicity and everything, and it didn't. But it was in the ADPKD form. So I would need to look and do this study more carefully, or I should say carefully in ARPKD to be very affirmative about it. But I wanted to go further in the adult because we can only test an adult. This is kind of the protocol people do. They do an adult before doing in a growing child.
Absolutely. And the one other question I have from the registrants ahead of time that you haven't already addressed is in the news, we're hearing more recently about drugs like Ozempic or other drugs that seem to be promising for CKD. Do those kinds of treatments translate to be effective for people with PKD?
Well, I must say, I don't know. I've not tested, and I don't know of anybody that have tested. I'm sure that probably York Pay mentioned about the dietary input maybe, or assays he's doing, and it's not the same thing. So I would not wet myself. He would be better than me to answer this question, and he probably looked into it. But I don't know about possibly, but I'm very careful because I'm always afraid it might have secondary and side effects which are specific to ADPKD and not the same than others. So I just cannot say it's negative, and I cannot say it's positive.
And I totally appreciate that. I will let the listeners know that we did do another session with Dr. Pei that Dr. Trudel has mentioned earlier today in the summit, and that video will also be available afterwards. So if there are people here who did not see Dr. Pei's presentation, he actually does answer that question in his presentation. So, and when this video ends up on YouTube, I will include the link to his video as well in the description. Well, that is for, we've got one more. Do we have anything else? No. Okay, so it looks like those are all the questions. And Dr. Trudel, I just want to thank you so much. We really appreciate your time, just because you have taken the time to make this information as easy as possible to understand, and that we really appreciate for the layperson, for the PKD patients and for their family members to feel some hope and be able to understand what you were talking about. I know that it's given hope to a lot of people who are listening, that there is potential somewhere down the road where there may be a treatment that can really make a big impact on PKD. So we're so thankful that you've joined us today.
It was a pleasure. It always is a pleasure.
That's great. I'll just let all of the viewers know that the video of this session, it will be available on our YouTube channel in the coming weeks. And all of the people who have registered will receive an email once the video is available. And I'll also say that if you have additional questions that you would like us to ask PKD researchers like Dr. Trudel, please feel free to email us anytime at endpkd@npkd.ca. The link or the email address and various links are in the chat right now. We are always happy to try and answer people's questions and to seek out wonderful advice like we received today from Dr. Trudel. So thank everyone for joining us today. Dr. Trudel, again, thank you so much, and wish everyone a good day.
Goodbye to everyone. Bye-bye.